Hello, students. Today, let's learn about photosynthesis. Photosynthesis is the process by which green plants, algae, and certain bacteria convert light energy from sunlight into chemical energy to form glucose. The reaction can be written as carbon dioxide plus water gives glucose and oxygen in the presence of sunlight. Here is the balanced chemical reaction of photosynthesis. Now let's discuss about the site of photosynthesis, the chloroplast. A typical mesophyll cell will contain approximately 30 to 40 chloroplasts. Each chloroplast is a double membrane organelle filled with a dense fluid called the stroma. Within the stroma lies a third membrane system composed of sac-like structures called thylakoids, which separate the stroma from the internal thylakoid space. In certain areas, these thylakoid sacs are arranged in stacks known as grana. Chloroplasts contain several pigments within their thylakoids that play a crucial role in the light reactions by absorbing different wavelengths of light. The main pigments include chlorophyll A, the key light-capturing pigment, chlorophyll B, and carotenoids serving as accessory pigments. A graph that plots a pigment's light absorption against wavelength is known as an absorption spectrum. The absorption spectra of thylakoid pigments provide insights into the relative effectiveness of various wavelengths for photosynthesis, as only absorbed light can drive this process. The absorption spectrum of chlorophyll A indicates that violet, blue, and red light are most effective for photosynthesis, as these are well absorbed. Accessory pigments also absorb light at other wavelengths and transfer energy to chlorophyll A. These findings are supported by an action spectrum for photosynthesis, prepared by illuminating chloroplasts with the light of different colors and then plotting wavelength against photosynthetic rate. The action spectrum for photosynthesis was first demonstrated by Theodor W. Engelmann, a German botanist, in 1883. Photosynthesis can be divided into two stages: light-dependent reaction, or light reaction, and light-independent reactions, or Calvin cycle. In light reaction of photosynthesis, solar energy is transformed into chemical energy in the thylakoid membranes of chloroplasts of leaves. During this process, water is split, supplying electrons and hydrogen ions as well as releasing oxygen as a byproduct. Chlorophyll absorbs light, which drives the transfer of electrons and hydrogen ions from water to a NADP plus molecule. Additionally, the light reactions also produce ATP. The Calvin cycle, on the other hand, begins with carbon fixation, incorporating carbon dioxide from the air into organic molecules already present in the stroma of the chloroplast. The Calvin cycle then reduces the fixed carbon to carbohydrates by the addition of electrons. The reducing power and chemical energy for the Calvin cycle is provided by NADPH and ATP respectively, generated by light reactions. Thus, it is the Calvin cycle that makes sugar, but it can do so only with the help of the NADPH and ATP produced by the light reactions. Before we go into detail about the light reaction, let me introduce you to photosystem. A photosystem is composed of a reaction center complex surrounded by several light harvesting complexes. The reaction center complex is an organized association of proteins holding a special pair of chlorophyll A molecules and a primary electron acceptor, while the light harvesting complex consists of various pigment molecules such as chlorophyll A, chlorophyll B, and multiple carotenoids bound to proteins. It converts light energy to chemical energy, which will ultimately be used for the synthesis of sugar. The thylakoid membrane has two types of photosystems: photosystem 2, PS2, and photosystem 1, PS1. Now let's see how the two types of photosystems work together in using light energy to generate ATP and NADPH, the two main products of the light reactions. The light reaction starts with photon absorption. The light harvesting complex of PS2 absorbs light energy, exciting electrons into higher energy levels. As this electron falls back to its ground state, an electron in a nearby pigment molecule is simultaneously raised to an excited state. The process continues until the electron reaches the pair of chlorophyll A molecules in the PS2 reaction center complex. Finally, the electron from the reaction center chlorophyll pair is transferred to the primary electron acceptor. The next step is water splitting, or photolysis. A water molecule is split into two electrons, 2 hydrogen ions, and an oxygen atom. The electrons are supplied one by one to the chlorophyll pair, each electron replacing the electron transferred to the primary electron acceptor earlier. The H+ ions are released into the interior of the thylakoid. The oxygen atom immediately combines with another oxygen atom generated by the splitting of a water molecule, forming an oxygen molecule. Now, in the following step, each photoexcited electron passes from the primary electron acceptor of photosystem 2 to photosystem 1 via an electron transport chain. The electron transport chain between PS2 and PS1 is made up of the electron carrier plastoquinone, a cytochrome complex, and a protein called plastocyanin. Each component carries out redox reactions as electrons flow down the electron transport chain, releasing free energy that is used to pump H+ into the thylakoid space, contributing to a proton gradient across the thylakoid membrane. Next, let's discuss the photon absorption in PS1. As seen in the photosystem 2, light-harvesting complex pigments use light energy to excite electrons and finally transfer the energy to the PS1 reaction center complex by exciting an electron of the pair of chlorophyll A molecules. The photoexcited electron is then transferred to PS1's primary electron acceptor. Subsequently, the photoexcited electrons are passed in a series of redox reactions from the primary electron acceptor of photosystem 1 down a second electron transport chain through the protein ferredoxin. Unlike the previous electron transport chain, this ETC does not create a proton gradient and thus does not produce ATP. The penultimate step is for NADPH synthesis. The enzyme NADP+ reductase catalyzes the transfer of electrons from ferredoxin to NADP+. 2 electrons are required for its reduction to NADPH. This process also removes an H+ from the stroma. The final step is ATP synthesis. Protons flow back into the stroma through ATP synthase, driving the conversion of ADP into ATP. Next, let me explain the stages involved in Calvin's cycle. The Calvin cycle starts with carboxylation phase. The enzyme ribulose bisphosphate. Carboxylase, oxygenase, Rubisco, facilitates the fixation of carbon dioxide into 5-carbon sugar called ribulose bisphosphate, RUBP, resulting in the formation of 2 molecules of 3-phosphoglycerate, 3-PGA. Next is the reduction phase. This process uses ATP for phosphorylation and NADPH for reduction, transforming 3-phosphoglycerate into glyceraldehyde 3-phosphate, G3P. Each cycle uses 1 G3P for glucose synthesis, meaning 1 glucose molecule is formed after 6 carbon dioxide molecules are fixed across 6 cycles. The third or the last step is the regeneration of RUBP. 5 G3P molecules regenerate RUBP, allowing the cycle to continue. 3 molecules of ATP are used up in this process.
Follow and subscribe us for more videos.